Assalam Alaikum students, welcome to the Bright Future Academy. Today we will discuss the Watson-Crick Model of DNA. It's also known as the Model of DNA or Watson-Crick Model of DNA. So अगर आप चैनल पे न्यू हैं, press the bell icon, do subscribe the channel, like and share the videos and give your feedback in the comment section below. Before studying the Watson-Crick model of DNA, you must have to know that what is DNA. DNA or deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Almost every cell in a person's body has the same DNA. DNA is the information molecule. It stores instructions for making other larger molecules called proteins and distributes it among 46 long structures called chromosomes. Chromosomes are made up of genes, which are the short segments of DNA, and each chromosome carries its own specific DNA. In this way, DNA stores all the genetic information, all the hereditary information, and it's an information molecule and a genetic material. DNA is located in a cell's nucleus, and almost every cell has the same DNA. DNA contains all the information, genetic information, hereditary information, all the information from embryonic development or from forming gametes to the new individual. So what is DNA? DNA is a genetic material which stores all the body's information. So how it performs its functions and where it's located? It's located in the cell's nucleus. It's an important board long question and you must have to draw its diagram in your paper. So how perform its function and who discovered the Watson-Crick model of DNA? In 1953, James Watson and Francis Crick proposed the structure for DNA. James Watson and Francis Crick both were colleagues and they discovered the structure for DNA while they were studying at a university. According to the Watson-Crick model, DNA molecule consists of two polynucleotide strands. DNA is a larger molecule and it comprises of two polynucleotide strands. Strands means a long fiber-like structure and poly means many. It means DNA contains two strands that's made up of polynucleotides. These strands are coiled around each other in the form of double helix. So what is nucleotide? Because nucleotides are present in the DNA structure and forms the DNA structure. Nucleotide is made up of three major components like phosphate group, pentose sugar, nitrogenous base. A phosphate group having a negative charge on it and a 5-carbon sugar that's known as pentose sugar. Penta means 5 and that's why it's known as pentose sugar, 5-carbon sugar, and nitrogenous bases. Phosphate group has a negative charge on it and it's said that DNA molecule as a whole is negatively charged molecule. The negative charge of DNA is due to the phosphate group. So these nucleotides are present in the DNA double helix and forms its structure. There is a phosphate sugar backbone on the outside of double helix. As shown in the diagram, phosphate sugar backbone is located at the onside of double helix. It means phosphate group and sugar molecules forms linkages among itself and located on the outside of DNA double helix. In a nucleotide, three major components like phosphate group, pentose sugar and nitrogenous base, from which two major things, phosphate group and sugar molecules, are on the outside of double helix and the remaining one, nitrogenous bases, are on the inside. As shown in this diagram, nitrogenous bases are located in the inside of DNA double helix. In double helix, the nitrogenous bases of opposite nucleotides forms pairs through hydrogen bonds. These nitrogenous bases forms linkage through each other by making hydrogen bonds, because hydrogen bonds is relatively much stronger bond and is forms where hydrogen is involved in the molecules. The important thing is each nitrogenous base forms pairs with the opposing nucleotides nitrogenous bases. This pairing is very specific. Each nitrogenous base forms pair with its nitrogenous base of opposing nucleotides and as the same over there. The nitrogenous base adenine of one nucleotide forms pair with the thymine. There are 4 nitrogenous bases that are present in the DNA double helix, namely adenine, guanine, thymine, and cytosine. Adenine forms pair with thymine of opposing nucleotide. As shown in this diagram, adenine forms pairs with thymine with the double hydrogen bonds, and cytosine forms pair with guanine. You can remember this by making a trick like mnemonics at GC. A adenine pairs with T thymine at GC. G guanine forms pair with C cytosine at GC, at Government College, or at GC Faisalabad. There are 2 hydrogen bonds between adenine and thymine as shown in this diagram. Here's 2 hydrogen bonds between adenine and thymine, and there are 3 hydrogen bonds between cytosine and guanine. Cytosine forms 3 hydrogen bonds with the guanine. So you can remember easily it by making mnemonics like at GC, at Government College. Adenine with thymine and guanine with cytosine. So in this way, the structure of DNA is formed or nucleotides as present in the DNA molecule. Hope you would like the video. If you like this, do subscribe, like and share the videos, and give your feedback in the comment section below. Stay tuned, keep watching.